Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event, which is titled Sex, Drugs, and Protocol, How Researcher Choices Impact Experimental Outcomes in Preclinical Diabetes Research. This webinar has been sponsored by DSI, a division of Harvard Bioscience, so a big thanks to them for making this event possible. Today, we're joined by Dr. Aileen King, Senior Lecturer in the School of Life Course Sciences at King's College London. Dr. King will present data showing how small alterations to experimental protocol and choice of model can substantially impact both welfare and data interpretation when studying blood glucose homeostasis in mice. And with that, I'm very pleased to introduce Dr. Aileen King. Aileen, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam, and thanks for the introduction. So, as Liam said, my name's Aileen, and I'm based at King's College in London. My talk today is titled Sex, Drugs and Protocol, and this is a study in which we use continuous glucose monitoring in mice to understand more about different factors which could influence outcome in preclinical diabetes research. In part one of this seminar, I'll discuss sex and how female mice differ from males with regard to blood glucose concentrations and variability. In the second part of this webinar, I'll focus on how variations in protocols can affect experimental outcome. And finally, in the third part, I will discuss whether changing experimental protocol affects our ability to detect drug effects. I'm going to start briefly by outlining blood glucose homeostasis for those of you who may not be in the field. Blood glucose homeostasis is primarily controlled from the pancreas. Clusters of cells known as islets of Langerhans contain beta cells which secrete insulin. Insulin promotes the uptake and storage of glucose from the blood into peripheral tissues such as the liver, the muscle, and the fat. Alpha cells within the islets produce glucagon, which promotes glucose production from the liver. And together, these hormones work in balance to maintain blood glucose concentrations within the range of around 4 to 7 millimolar in humans. Diabetes develops due to an absolute or relative deficiency of insulin, and the main symptom of diabetes is hyperglycemia. Animal models are an important tool in diabetes research, and their use dates back to the discovery of insulin using Marjorie the dog 100 years ago. Nowadays, the most common species used in diabetes research is the mouse. In similarity to other areas of research, female mice are underused in preclinical diabetes research. There is an assumption that the estrous cycle will lead to variability. The estrous cycle is the 4 to 5 day long reproductive cycle of the mouse, and it's equivalent to the menstrual cycle in humans. It's characterized by fluctuation in sex hormones and is split into stages depending on estrogen levels. Proestrus and estrus have high levels of estrogen, and metaestrus and diestrus have low levels of estrogen. This assumption that it will impact on results stems from the fact that estrogen has known effects on beta cell biology. Another reason females may be excluded from a lot of preclinical diabetes research is that males tend to have a more hyperglycemic phenotype, and it's been questioned whether that's more suitable to test drugs. There are several ways to assess blood glucose homeostasis in mice. The most common way, perhaps, is to measure random blood glucose measurements at fixed single time point measurements. This requires handling and pricking the end of the tail of the mouse to create a small blood sample. As you can see from this video, a needle is used to make a small prick in the tail and a very small blood sample. We need less than 1 microliter is used with a normal glucometer. We get the answer in a few seconds of what the blood glucose is, and as you saw, the mouse is only partially restrained. Another really common method in diabetes research is glucose tolerance tests, and this is both used clinically and with animal models. This assesses glucose homeostasis under stimulated conditions. The mice are fasted to get a basal blood glucose concentration, and then a glucose bolus is administered. This technique requires multiple handling points to measure the blood glucose, as well as the administration of the glucose. Continuous glucose monitoring is a relative new technology in mice, although it's been used in humans for many years. In this study, we have used this where we implant glucose probes in the aortic arch with a transmitter placed subcutaneously. This allows blood glucose concentrations to be transmitted by radiotelemetry to a nearby computer. The cage is placed on a telemetry mat and mice remain unrestrained. We have collected data every 10 seconds, providing 8,640 measurements a day in comparison to glucometer measurements, where we usually only measured once a day. This has allowed us to measure glycemic variability, which is important in diabetes research, as increased variation in blood glucose concentrations can be associated with diabetic complications. It also gives us detailed information on the mean, the median, target ranges, and allows us to investigate the effect of common in vivo techniques. This video is just to show you that the mouse, after it's recovered from the surgery, just wanders around completely normally in its cage while we're measuring blood glucose. So it means we can measure blood glucose concentrations without interacting with the mouse. The potential benefits of continuous glucose monitoring are both on the welfare side, that after the surgery, we are studying conscious animals in a normal state. There's minimized human presence and therefore most likely stress reduction. Finally, because we've got access to blood glucose concentrations at all time, reduced blood sampling is required. Scientifically, it captures data that otherwise would be missed. This includes glucose excursions and nighttime readings. We can also use this technology to study the impact of researcher intervention. So the aims of this study were to establish what normal blood glucose concentrations were in C57 black 6 mice. We chose this strain as the most commonly used strain in research. We also wanted to know how much the blood glucose concentrations were fluctuating, as you can't tell that from a single time point measurement. We wanted to ascertain whether female mice do indeed have more variable blood glucose concentrations due to estrogen. And we wanted to see what the impact was of varying experimental protocol would have on different outcomes. Finally, we wanted to assess whether higher blood glucose concentrations are required to detect drug effects. So what are normal blood glucose concentrations in mice? As I just mentioned, these are for C57 black 6 mice, and it may very well change between different strains. So in the males, we found that the median blood glucose concentration in undisturbed mice was 6.9 millimolar. And for those of you in America, this is equivalent to 125 milligrams per deciliter. As you can see from the graph, blood glucose concentrations are continuously fluctuating. We therefore set a target range that was 25% above the median and 25% below the median. And this is based on something that's done in humans. This allows us to assess what a normal target range would be. In this case, 5.2 to 8.7 millimolar. And this encompassed 90% of all blood glucose readings measured in the males. In the females, the median was 6.1 millimolar. This is around 110 milligrams per deciliter. The normal target range in females was between 4.6 to 7.6 millimolar, and this was calculated the same way as 25% above and below the median. In females, this encompassed 95% of all blood glucose measurements. Perhaps you can see here, there's already some evidence showing that the females are not as variable as the males. So By measuring the blood glucose accurately over very many different time points, we could ascertain that indeed male mice have got a higher blood glucose concentration than females. And this was irrespective of whether estrogen levels were high, which is seen by the proestrus and estrus stage, designated as PE in this graph, or if estrogen is low, which is designated by the metaestrus and diestrus stages, which is shown by MD. This is true during the day and during the night. In male mice, the blood glucose was higher at night. This isn't surprising because mice eat about 2/3 of their caloric intake during the night. We didn't see this significantly in females, although there was a slight trend. So when it comes to measuring glucose variability, there's several different ways to doing this. We used initially 3 different ways. The first way looks at every single data point and how far it varies from the median. The second way looks at values 2 hours apart, so it's more of a rolling over the day variable. And the third day specifically looks at larger spikes in blood glucose, and these are spikes that are 1 standard deviation above or below the median. Irrespective of which method we used to quantify variability, we found that the males always had far more variability than the females, irrespective of estrus stage. I'll just concentrate on one so you can have a closer look. So here you can see that both during the day and during the night, males nearly have twice as much variability in the blood glucose concentrations than females, and this was irrespective of whether the females were in the proestrus estrus state or the metaestrus diestrus state. We also wanted to look what I described earlier was how much variability there is, for example, within a day. But we also wanted to look at between day variability, and you would specifically expect this to change if estrus was having a large effect on the females, because they should have, in that case, different blood glucose on different days. However, as we saw before, the male mice had more day-to-day variability in the blood glucose than females. Finally, we decided to look at how much variability there was between animals, and to do this, we looked at how much individual mice deviated from the cohort mean. And again, in the males, we found the coefficient of variation around 10%, whereas in females, it was only 10% of females and 15% in males. And again, this reached statistical significance in males. So the take-home message from this part of the talk is that female mice have lower blood glucose concentration than males. Female mice also have less variable blood glucose concentration than males. The estrus cycle doesn't lead to significant changes in blood glucose concentrations. We then wanted to go on and look at how common procedures affected blood glucose concentrations. And the first thing we looked at was a normal husbandry cage change. And we found that when the cage is changed, the blood glucose increases for up to 2 hours in both males and females, with an increase in blood glucose of around 1 to 2 millimolar. To look at this further, we decided to investigate specific in vivo procedures that we used as part of our research. The first thing we looked at was just a general disturbance. This is when the researcher or a member or a lab technician walks into the animal room and perhaps checks the welfare. It involves the researcher lifting the lid but not touching the mice. We then looked at the effects of handling, the effects of measuring blood glucose, as I previously showed you, and finally, the impact of an intraperitoneal injection. So in both males and in females, Just merely walking into the room and removing the lid from the cage increases blood glucose in both males and females by between 0.5 and 1 millimolar. This is about 18 milligrams per deciliter. In the males, handling then increased the blood glucose concentration further, and in fact there wasn't much difference between handling and the blood sampling. So by the time you've handled the mouse, the blood glucose has already increased. As you can see from the large error bars, this is quite variable between different mice and males. And by the time you've restrained the mouse for an IP injection, the blood glucose has increased by around 2 millimolar. In females, we see a similar pattern. This effect of handling and blood sampling is not quite as high, but by the time you restrain the mouse for an IP injection, the blood glucose increases by about 2 millimolar. So I'll now discuss the glucose tolerance test protocol, which is one of the more widely used protocols in preclinical diabetes research. The first stage is usually fasting. This can happen 6 hours before or 16 hours before if an overnight fast is taking place. And the reason we fast is to ensure a stable blood glucose and reduce variability, making sure that the blood glucose concentrations aren't depending on what the mouse has been eating. Just before the start of the GTT, the researcher will quite often enter the room to weigh the animals to determine their glucose dose. Even if this is done previously, the researcher does have to enter the room to measure the blood glucose immediately prior to the start of the glucose tolerance test. And this gives the baseline glucose value as a starting point. The start of the GTT then occurs when glucose is administered, and then, as I mentioned before, blood glucose is then measured at regular time points after this. This graph just shows that you can see the slight impact of the researcher intervention. Here you can see that the researcher has entered the room to start the fast, and you can see there's a slight increase in blood glucose as that happens. At the end of the fast, just before the glucose tolerance test, again the researcher enters the room and the blood glucose increases somewhat. And of course, as expected, when you inject glucose, the blood glucose increases substantially. And finally, you can see an impact of the researcher entering the room to feed the mice to end their fast. So the increase in blood glucose concentration seen during a glucose tolerance test is partially due to the effects of the procedure itself. So in grey, you can see in both males and females what their baseline glucose concentrations were. And then you can see the accumulated effect of entering the room, handling the mice, and doing the injection. In yellow, you can see what the actual effect of the glucose bolus is. So around 2 millimolar of the effect you see is actually due to the procedure itself. So we wondered whether altering the GTT protocol could affect welfare and/or outcome. Now, one of the things we decided to look at was fasting. And although it might seem quite obvious how you remove the food, I've realised through working in different laboratories that how this is done actually differs between labs and is very rarely reported in the literature. We also wanted to look closer at how long we should fast for. Finally, we wanted to check what differences were made by differing the glucose administration route. So to initiate the fast, we've looked at different methods. One method is actually just to put the mouse into a new cage. This ensures that there's absolutely no food remnants in the cage that the mouse can eat. However, it may be associated with a stress response. To try and prevent the stress response, you can carry out a method called bedding retention. In this case, the mouse is transferred to the new cage, as I said before, but this time you take the old bedding from its old cage, shake it out to ensure there's no food in there, and put it in the new cage. And this adds familiar smells, which helps reduce stress in the mouse. Finally, perhaps the most obvious way of initiating a fast is just by removing the food hopper in the lid. And in doing this, the mouse remains in the original cage. However, there is the chance that there may be food remnants at the bottom, especially when you're using a high-fat diet, which is more crumbly in nature. So counterintuitively, of course, we're fasting the animals to decrease their blood glucose, but the first thing that happens actually is the blood glucose increase, and this is highest when you do a whole cage change, seen here in red. So the other cage changes still have this effect, but to a lesser extent. This is the whole fast, where to the left of the graph, you can see that spike in blood glucose, which is the researcher intervention. And this effect of the researcher entering the room and starting the fast lasts around 1.5 to 2 hours. And it's only then that the animal actually starts to have blood glucose that is at least or at or below the original basal blood glucose concentrations. As you can see in the yellow line, this is an overnight fast, and the blood glucose dropped quite considerably. But this will be missed because the extent of this will be missed under normal circumstances, because as soon as the researcher enters the room again and measures blood glucose, the blood glucose increases again. So we wanted to look at what the most effective fast was, and this graph is a little busy, so I'll just simplify it to start with. So if we start with the yellow small bars, this is ad libitum fed animals that have just been fed as normal. Now, if you cast your mind back to our normal target ranges, you can see that about 5% of the time, the blood glucose of the mouse dips below one of these normal target ranges. And we can see that here in both males and in females, irrespective of whether they're in proestrus, estrus stage of the estrus cycle or metestrus, diestrus stage. And if we fast the mice overnight, once we've got past that initial 2 hours where there is the effect of the researcher intervention, then blood glucose indeed does dip 100% of the time below the baseline target range. At the other end of the scale, we have what happens if you don't change the cage at all. And in this scenario, in males, only 30% of the time the animals are below their target range. And in females, this is around 50% of the time. And other cage changes lie somewhere in between. So from this, we could see that the most effective cage change fast was 16 hours, and the least effective fast was 6 hours with no cage change. However, we were worried that the mice really were going quite low when we did the overnight fast. And again, if we concentrate on the white bars to start with, you can see the minimum blood glucose that the mice will normally meet under normal conditions when they're fed. So this is the lower limit of what normal is. And as you can see, in both males and females, it's just below the 5 millimolar rate. In animals that are fasted overnight, this drops substantially. Now, unfortunately, they drop so far that they're actually below the threshold of what we can accurately measure with our DSI probes, which have a lower limit of 2.8 millimolar, which is 50 milligrams per deciliter. So we can't say precisely how low they go, but they do seem to go below this, as you can see here. And we feel this is unacceptably hypoglycemic and stressful for the mice. In all 6-hour fasts, the mice, despite the fact that they're lower on the low end of normal, they never actually go hypoglycemic. So we have found that alterations to the fasting protocol can affect the efficiency of the fast, and it can also affect animal welfare. However, as scientists, one question should always be, will it affect the outcome of what we're trying to study? So we found that when the male mice were fasted overnight, as you can see here in yellow, they had impaired glucose tolerance. This means that when the mice were fasted overnight prior to the start of the glucose tolerance test, they were slower at removing glucose from their blood. This was also partially seen with the most stressful of the cage changes, which is the whole cage change, albeit this effect was much more mild. In female mice, if you used a 6-hour fast with bedding retention, there was no difference between the proestrus and estrus stage of the estrus cycle versus the metestrus and diestrus stage, and you got very similar results. However, if you had fasted the female mice overnight, you found that the metestrus diestrus mice had slightly impaired glucose tolerance. We found a similar scenario with the more stressful of the cage changes, which is the whole cage change. And here you could see again that if you changed the whole cage prior to a 6-hour fast, you suddenly saw a difference depending on the estrus cycle, with the metestrus diestrus animals having slightly more impaired glucose tolerance. So the take-home message here is that overnight fasting is not recommended on welfare grounds. Now, we realise a lot of you out there already know this, and a lot of researchers are already using a 6-hour fast. But we did a short review of the literature a couple of years ago, and we found that 60% of all papers were still using overnight fasts in glucose tolerance tests. Now, there has been, this has improved. 10 years ago, only about 20% were using a 6-hour fast, and now about 30% are using a 6-hour fast. So we are seeing improvements as researchers become more aware, but we're still not quite there yet. We should be worried about this because it potentially impacts the results in both male and female mice. The whole cage change is not recommended on welfare grounds because we know that they get stressed by this. However, we do know that they get stressed by any kind of intervention, but the whole cage change is not necessary and could impact on results. We therefore recommend the 6-hour fast with bedding retention cage change. We find this is the most efficient fasting. We've got data on that I haven't had time to show you today, and also it improves welfare compared with just changing the cage without retaining the bedding. So finally, we wanted to investigate whether we could further refine the glucose tolerance test. So far I've discussed that we should perhaps be using 6 hours rather than 16-hour fasts. And if you're going to change the cage at the start of the fast, you should use bedding retention. But what about the glucose administration? Because the most commonly used way to administer the glucose is by an intraperitoneal injection, which obviously requires restraint. So mice can be trained to voluntarily ingest glucogels or gels containing drugs. You can mix the glucose with gelatin and flavouring, or you can mix a drug with gelatin and flavouring. And especially with the drugs, it's good to use a non-sugar based flavouring, such as non-sugar chocolate flavouring, which the mice particularly like. You present this to the mice on at least 3 occasions prior to starting a glucose tolerance test, and they soon get used to this and eat it. And we deem them trained when they eat it in less than 60 seconds because we think this is fast enough that they are getting a real glucose bolus. So the implications of mice voluntarily eating glucose gels is that it could be more physiologically accurate. This is because oral glucose initiates the incretin system, and this is when gut hormones are released in response to oral glucose, and this induces higher insulin secretion. Arguably, it possibly induces less stress. There's no restraint required if you just present the mice with the glucose gel, which it voluntarily eats. However, temporary separation is required to make sure the right mouse eats the right glucose gel and they don't steal from each other. So we found that voluntary gel ingestion reduces glucose peak. Now, in these experiments, we have compared to oral gavage because it still means that the glucose is administered orally, but the animals are restrained in a similar fashion to an IP injection. When the gels were consumed voluntarily, we see a reduction in the glucose peak in all males and in female mice. So this led us to ask the question, do we actually need this elevated peak in glucose to detect drug effects? Now, if you cast your mind back to perhaps what we might regard as our more severe protocol is when we overnight fast males, and we have a peak of glucose at 25 millimolar. In our most refined glucose tolerance test, which is in females where we've used a 6-hour fast with bedding retention and voluntary glucose injection, the peak is only at 12 millimolar. And the question is, does it matter that your peak is only half of that is seen in the male experiment? So to do this, we made metformin into gels and used a non-sugar sweetener to flavour it with chocolate, and this was presented to the mice 30 minutes before the glucose tolerance test, and then the glucose tolerance test was also a voluntary ingestion of a gel. And we saw in males that as you can see in the control group, the peak is only 15 millimolar, but despite the slower peak, there's a very clear effect of metformin. Metformin is the most commonly prescribed drug for type 2 diabetes, so we thought this was very clinically applicable. We saw the same in females, and again, as you can see in females in the control, the peak is only around 11 millimolar, but we can still see very clear effects of metformin. And this indicates that perhaps you don't need to be aiming for this massive increase in blood glucose during the glucose tolerance test. So the take-home message from this part is that refined protocols can be used to detect drug effects despite blunted glucose responses in the control. So the summary of the talk is that female mice have lower blood glucose concentrations, but also lower variability. We've looked at that regarding sample size calculations, and although the effect of the drug is often smaller in females, the lower variability means that you need the same number of mice as you would do in the males. Estrus does not affect random blood glucose concentrations, and it doesn't affect glucose tolerance when using the most refined protocols. But you should note that estrus could start having an effect if the mice are a little bit more stressed. Fasting mice overnight is not only a welfare concern, but can also impact results. And elevated glucose responses are not required to detect drug effects. And this again just feeds into that perhaps it's just as valid to use female mice with their lower blood glucose levels. In conclusion, female mice should be included in studies of blood glucose homeostasis. Subtle alterations to protocols can affect results. And I think the most important thing is to be consistent, to be aware that small differences can affect results. So you've got to keep consistent in what you do and report protocols accurately so people reading your papers know exactly what you've done. I just want to acknowledge my PhD student, Matilda Kennard, who did all this work. Meenakshi Nandi, who was the expert in telemetry and co-supervises Matilda, DSI for all their support and help, and the British Pharmacology Society for funding Tilly's PhD studentship. Thank you.
Fantastic. Well, thank you, Aileen, for a really great presentation. And with that, we'll move right on to the Q&A. All right, let's jump right in. So Aileen, first question here. Does continuous glucose monitoring tell you something that you can't measure with normal glucometers?
Thanks, Liam. Yeah, it depends very much on which experiments you're doing and what you want to find out. The most obvious things that a glucometer can't measure is fluctuations in blood glucose, and this happens all the time. Luckily, we've actually looked at how accurate the glucometer measures glucose tolerance and glucose tolerance test. And the good news for those who don't want to do telemetry is the accuracy of glucometers in the glucose tolerance test is pretty good. However, you do miss subtle drug effects when you're measuring with a glucometer. So we've studied different drugs. We've studied metformin and exendin 4 using different concentrations of drugs. And in some cases, you can see differences with the continuous glucose monitoring that you can't see with the glucometer. And also when you administer the drug, what you can do with the continuous glucose monitoring is nearly see exactly how long it remains in the system and how long that effect has. And finally, it depends if the mouse is doing something very different in the day versus during the night. And this happens particularly when the animal is particularly diabetic. So we've done some transplantation experiments in strepsitosin animals, which we made diabetic by injecting the drug strepsitosin. And then we transplanted islets of Langerhans. And what we see using a glucometer was that you could measure blood glucose and it looked like the animal was cured with a blood glucose of less than 11.1 millimolar. However, when we used continuous glucose monitoring, we realized that the mice were very hyperglycemic during the night, and we missed this with a normal glucometer. So especially if the animals are overtly diabetic, it can be useful to use continuous glucose monitoring. But just it's very good to find very subtle effects and also to see what's happening when you're not touching the animal.
Fantastic, great answer. Next question here. So it's known that males are larger than female mice. Have you taken the body weight of the mice into account? And if so, what are the sort of implications of that on the data?
I mean, I think, yeah, in some cases, we've actually done this also with high-fat diets. Even when you take into consideration the size of the mouse, the blood glucose does tend to be higher in males. Normally, we've looked at also at mice at different ages, and we don't see really enormous effects from, say, a mouse that's about 10 to 12 weeks versus a mouse that's around 24 weeks. But the effect of age is, again, another interesting question, which automatically has the effect of the mouse being slightly bigger. But in general, we haven't really looked in detail about whether the size of the mouse is affecting it. I guess we could do that correlation, but the blood glucose is probably fluctuating so much that you can't determine. It's not as simple as the heaviest mouse has always the highest blood glucose.
Fantastic. Next question here. So with regards to blood glucose increasing in the in vivo procedures, did you look into changes following oral gavage as well? Can you just clarify that a bit?
Yes. Sorry, I didn't clarify that well. Yes. So the same thing happens. I think especially in the male mice, nearly as soon as you pick them up, it nearly doesn't matter what happens after that. They have this sort of 2 millimolar increase in blood glucose. So there is definitely a stress effect in the oral gavage as well. And there will be a slight stress effect even by administering the voluntary glucose to a certain extent, because as soon as you walk in the room, the blood glucose increases. And I think it's just something we can't get around. You can't generally do experiments without ever being there. So I think it's more just being aware how different aspects can affect, and just being aware that if you do something slightly different from normal, it really could impact. So you've got to make sure you've always got the right controls. We've even seen an impact of a different researcher doing the experiment, especially if the researcher is obviously a little bit less experienced. So you just need to keep things, I think you need to accept that this is a phenomenon that we've just got to accept, because there's nothing we can do about it, but just keep everything absolutely as consistent as possible.
Yeah, that's a really great point, and important to keep in mind for sure. All right, so next question here. What was the age of the mice that they used? And maybe I can broaden that to how might age affect the results here?
Right, so the age that we used, we implanted, sorry, I should have mentioned this during the talk, we implanted the probes at around 10 weeks in the experiments I've shown you today. This is because the probes can't be placed in mice that are too light. So we always had to have the mice at least 25 grams or 23 grams and above. We have actually done a study where we have carried out high-fat feeding, and for that we needed the age-matched control. And obviously when we're feeding the animals for 8 weeks, during that time they also age. So what we see is that they still react pretty much to everything you do, but the older mice, say if we look at mice that are between 20 and 24 weeks, their response is a little blunt compared to mice that are about 10 to 12 weeks old. So there is a slight effect of age that the mice, you can see just a slight blunting of response, but the response is still there.
Fantastic. All right, here's a good question here. Do the differences in resting glucose and glucose variability in males and females reflect sex differences in insulin secretion or meal patterning?
That is a good question. I mean, there has been studies showing that insulin secretion can be higher in females, but again, there's not an awful lot of studies in that. These probes also look at activity, and we did look at activity levels between males and females, and it gets a little bit complicated in mice, because obviously if you're moving, your blood glucose could actually go down because you're using glucose. But in the scenario of a mouse, the reason they might be moving is to get up and get some food. So we actually found that the females were moving more than the males. So we're not sure exactly why that is. So to answer the question, it's also quite established that males tend to have higher insulin resistance than females, so it might be to do with that as well. And in fact, when we've done insulin tolerance tests, we do find that the males have worse insulin tolerance than females, so they're not as sensitive to insulin. So it's probably a combination of several factors.
All right, great answer. All right, next question here. Thank you for the fantastic data. I was wondering whether the mice were handled prior to the studies performed in order to get accustomed to the researcher presence and interaction and scruffing.
Yes, we were handling the mice before the probes were implanted. So we have tried to look at whether there's any kind of habituation. And what we did find is the first glucose tolerance test you do is always impacting the mouse the most. So once you've done the first glucose tolerance test, the next glucose tolerance test doesn't go as high, but any others after that are all the same. So there is what I call an effect of the primary glucose tolerance test, and then after that they seem to not really habituate. So there's a tiny bit of habituation from the first time you meet, you kind of interact with them. Now, this could be really important because, for example, if you did a glucose tolerance test and then gave a drug, and then at the end gave another glucose tolerance test, you'll inevitably find that the second one is the animals are have better glucose tolerance. So it's obviously, as you all know, it's important to have the proper controls, but it is we would recommend that you have some kind of mock-up of a glucose tolerance test before your first one, just to habituate them slightly. But we were a bit surprised. They never seemed to get used to the cage changing. Now, of course, we don't really know what's happening before we put in the probes when the mice are younger, and maybe their response is even higher at that stage. But their cage is changed every week by experienced members of staff, and they still seem to increase in blood glucose to it. So there are some aspects of habituation, but at the end of the day, they still react to a lot of things. As I mentioned before, when we use slightly older mice, they reacted slightly less. And again, that indicates there might be a very, very slight habituation. But in general, you can't assume that just by handling the mice a few times that the mouse isn't going to react when you go to do your experiment.
All right, interesting. All right, so here's a question. Are rats affected? Do you think rats might be affected in the same way as mice?
I actually did a glucose, continuous glucose monitoring study in rats, but I wasn't particularly looking at research or intervention to the same extent. So I'm not such an expert in rats. I don't use them much in my own research, apart from that one study that I did. I guess you may be able to habituate rats a little bit more. They seem, at least when you go into an animal department, they're not quite as jumpy as mice, but that might just be my perception. So to be honest, I'm not really sure what the responses that rats have. One thing I did notice about when I did a continuous glucose monitoring rats is their circadian rhythms were far more pronounced. They really did have very, very clear differences between day and night in normal glycemia and also in the diabetic state.
Interesting. Well, it might be interesting if somebody can look into that, maybe into rice or into rats. Sorry. All right. So next question here. So does the day of the ovulatory cycle not matter in a protocol that identifies glucose homeostasis compared to a diet-induced obesity group?
Yeah, sorry, I'm just trying to think. We did also look at high fat fed female mice, but we don't have such extensive data in it. I'm not entirely sure. These are all normal glycemic animals that I showed you today. So it depends whether the sort of the high fat diet interacts with the estrus stage. In general, we have found in these mice that estrus only has an effect if the animals are particularly stressed. So we've done other experiments that I haven't shown you today. For example, if you transport the cage to another room to do your glucose tolerance test, that only affected female mice the estrous stage if you had a whole cage change or you'd overnight fasted them. So I think with regard to female mice, if you've got refined protocols, in our experience, the estrous stage doesn't impact on results. But if the mice are feeling a bit more stressed, then the mice that are in the metaestrus diestrus stage are more likely to have higher blood glucose concentrations. But I can't answer directly on what happens in the high-fat fed mice. I think we ended up with so few animals in that in the end that I don't think we were able to look into a lot of detail. So I'm not entirely sure if how that corresponds in the high-fat diet model.
All right, excellent. All right, so this question kind of goes back to insulin secretion versus feeding behavior. But do you know how insulin or the levels of insulin secretion and how they might be affected by either the 6 or 16-hour fast?
I mean, in theory, I know. The quick answer is no. I'm not entirely sure. I presume insulin secretion should reduce with a fast because you don't have elevated blood glucose levels in your blood. A lot of the stress effects actually comes through adrenaline and cortisol, and they also can impact on insulin secretion, but they can also impact on insulin sensitivity. So especially in the males, I presume that the reason that they've got impaired glucose tolerance when you've overnight fasted them is actually because they've got higher insulin resistance. And we haven't been able to measure that yet, but that's something to look into in the future.
Yeah, for sure. Very interesting. Um, do you think similar results would be observed using the refined protocol, but without continuous glucose monitoring?
Uh, yeah, we have been able to repeat some of these experiments using just a glucometer. As I said before, the one experiment that a glucometer is actually really quite accurate is the glucose tolerance test. So we've done glucose tolerance tests where we've just administered the glucose and then just walked away and left the mouse. And then we've also done it in glucose continuous monitored mice, where we still go and measure the blood glucose at 15 and 30 and 60 and 90 and 120 minutes. And in these circumstances, the glucometer is actually pretty much superimposing what we see if you don't use the glucometer at all. So I do think that this is relevant even if your animals don't have a telemetry device. And during these experiments, we actually, because another result actually I didn't mention is single housing, and it's known that single housing mice is not good for them. And we, during these experiments, you can only have one mouse on each telemetry mat. So in each cage, we had one mouse that had the telemetry device and one mouse that was just there to keep it company. And all experiments were also carried out in what we call these buddy mice. And we found very similar results. So yes, I do think it would be relevant to mice, even if they don't have telemetry devices.
Great answer. So going off of that, so considering stress, as you showed in your presentation, has a big impact on the glucose levels. Does the handling that's required for traditional glucose testing, did that have an effect? Do you think you said it was superimposing the data, but do you think that should have had an effect on the glucose levels measured?
I'm not sure quite understand. I mean, definitely measuring the blood glucose does increase the blood glucose. So yeah, you can tell that just by measuring the blood glucose over. So when I said that the median blood glucose was say 6.9 in millimolar, I'm guessing most people, when they measure blood glucose, and us included, if you're measuring blood glucose with a glucometer, you usually get a little bit higher than that. So I definitely think that the blood glucose you're measuring with a glucometer is a result of the mouse being slightly stressed at the time. But I'm fully aware we can't do anything about it. So we can't all do telemetry in every single mouse because that would be impractical. So I don't think we should be particularly concerned that measuring the blood glucose is increasing the blood glucose. I think we just need to be aware about it and be aware that if you're not used to handling the mice, say for example, you go away on holiday and you ask a student who doesn't do it very often, you could end up having an increased blood glucose just because of their inexperience. So I think my kind of overall take home message is that it does happen, but just be aware and do everything you can to make sure the mice are as stressed as little as possible, and just keep things consistent. And you should be able to work within that the blood glucose will be slightly increased just by handling the mice and measuring the blood glucose.
Fantastic. Yeah, some really great points. And I think we'll actually end the Q&A on the high note. So thanks so much, Aileen, for the really great insights today. It's been a real pleasure having you with us.
Thank you.
And a big thanks also to the audience here for joining us today. And we look forward to having you with us again next time. Have a great day, everyone.